Hi students, now today we are going to study new topic, that is genetic code. Now before proceeding with genetic code, you should know what is DNA. See, DNA is the master molecule. DNA is the master molecule of the cell. Means what? Means DNA is the master molecule, means DNA is only responsible for the formation of protein. What type of protein is going to synthesize in our body, in our cell? It is not at all decided by us. Who is deciding this? What type of protein is going to synthesize? It is decided by our DNA only. Okay, so DNA is nothing but it is the master molecule of the cell, means it is responsible for the formation of protein synthesis. This is the basic thing we should know. Now, next, in our next topic we are going to study about protein synthesis. So in short, you should know what is protein synthesis. See, synthesis means production. Okay, proteins production, that is called as protein synthesis. Actually, this protein synthesis is completed in two steps. One is called as transcription and another one is called as translation. Transcription means, see what happens when from DNA RNA is produced or RNA is synthesized, that step is called as transcription. And when from RNA protein is synthesized or produced, that process or step is called as translation. Okay, now what happens? One scientist was there. Name of the scientist was Crick. According to Crick, this protein synthesis is taking place in the form of coded language. To this coded language, another term was there, that is called as cryptogram. And that coded language is called as genetic code. And this genetic code is made up of, or it is made up of code word. Those code words are called as codons. See again what? According to Crick, according to scientist Crick, this protein synthesis is always taking place in the coded language. One certain coded language is there. That coded language is called as cryptogram, or it is also called as genetic code. And according to him, this genetic code is always made up of code word, and those code word are called as codons. Okay, that time previously in olden days, scientists were knowing, or they had this knowledge, that there are total 4 types of nucleotides in DNA: cytosine, adenine, guanine and thymine. And there are 4 types of nucleotides in DNA, and there are 20 amino acids which are required for the synthesis of protein. This knowledge was there with the scientist, that there are total 4 types of nucleotide in DNA, and from this 4 nucleotide there are 20 amino acids produced, and these 20 amino acids are responsible for the formation of protein. Now one scientist was there, very famous scientist, and his name is G. Gamow. He suggested that there must be the combination of 3 nucleotides. Total 4 nucleotides were there: adenine, guanine, cytosine and thymine. But what G. Gamow suggested, that there must be combination of 3 nucleotides, and these combination results into the formation of 64 amino acid. This I'll explain you with the help of diagram. See this. According to G. Gamow, there is combination of 3 nucleotides. Can you see this combination of 3 nucleotide? According to him, there is combination of 3 nucleotide, and they are responsible for the formation of 64 amino acid. Okay, now this was suggested by the scientist G. Gamow. Other scientist was there, 2 scientists were there, Nirenberg and Matthey. These 2 scientists have supported the theory which is suggested or given by G. Gamow. How they supported, I'll tell you. What they did, these 2 scientists, Nirenberg and Matthey, they prepared artificial mRNA. And mRNA is responsible for the formation of protein. But what little bit change they did, they prepared artificial mRNA, and with the help of artificial mRNA, here no, in mRNA they use only one type of new nitrogen base. Actually there are 4 types of nitrogen bases: adenine, guanine, cytosine, and as it is RNA, instead of thymine, uracil is there. So what they did, they made artificial mRNA using only one nitrogen base, that is uracil. Just consider whole full mRNA is there, only U will be there, only uracil is there. They found that this mRNA, which is containing only uracil, is responsible, or it produces polypeptide chain, that is polypeptide chain of phenylalanine. It means they supported that combination, yes, combination of 3 nucleotide results into the formation of nucleotide or amino acid. Then another scientist was there, Dr. Hargobind Khurana. Dr. Hargobind Khurana, again very much famous scientist. He also supported the theory of G. Gamow, that yes, there is combination of 3 nitrogen bases, or 1 codon is responsible, or 3 nucleotides are responsible for the formation of protein or amino acid. So how he supported, we'll see. See, Hargobind Khurana, no, he produced artificial DNA, synthetic DNA. From DNA he synthesized mRNA. But here in mRNA, what he did, he used the sequence of only 2 nucleotide, or in DNA synthesis also he used only 3 nucleotide. Can you see here? See, only 2 nucleotides are there: cytosine, uracil, cytosine, uracil, cytosine, uracil. Like this, he used only 2 to 3 nucleotide, and he found polypeptide chain which is containing 2 types of amino acid. One is leucine and another one is serine. See, it means this CUC, this one codon is responsible for the formation of leucine. This another codon, that is UCU, this second codon is responsible for the formation of serine. It means he also supported that yes, 3 nucleotides are responsible for formation of one amino acid. So they all supported the theory given by the scientist G. Gamow. Now we are going to study about characteristics of genetic code. Very important characteristics. 10 characteristics we have to study. How you will keep these characteristics in your mind? See, you will keep these keyword in your mind: CC, DDD, NN, UTI. CC, DDD, NN, UTI means the C for it is commaless. Next C for it is codon, it contains codon. D for it has distinct polarity. Next D for degeneracy. Next D for dual nature. N for non-ambiguous. This N for non-overlapping. U for universal. T for triplet code, and last I for initiation codon and termination codon. So this is the way in which you can keep this characteristics in your mind. So we'll proceed with first characteristic. See, genetic code is always commaless. Means what? We'll consider this is the genetic code. Here you can see the spaces or gap is there. That's why this is line. This is a sentence. But genetic code means you can just imagine here one more alphabet is there. Here one more alphabet is there. Here one more alphabet is there. All the 4 nucleotides, that is adenine, guanine, cytosine, thymine, they are continuously present here without any gap or without any punctuation mark. That's why it is called as genetic code is always commaless or without any gap or without any punctuation mark. Next characteristic is genetic code contains codon. Means what? Genetic code is always in the form of triplet. These triplets are called as codon. Now what is this triplet? Triplet means, suppose we will consider this is the genetic code. This GEN will be the first codon. ETI will be the another codon. Suppose here we have A, CAC will be the another codon. Okay, so genetic code is always in the form of triplet. Now codons are always present on mRNA, that is messenger RNA, and their anticodon, their opposite codons, are always present on tRNA, that is transfer RNA. Now third characteristics, genetic code has distinct polarity. Means what? Suppose this is genetic code, it has specific pole. This end is called as 5' end and this end is called as 3' end. So genetic code, see, if we are reading something, we will read in this direction only. If this is 5' end, this is 3' end, we will always read from 5' end to 3' end. Here same way genetic code is always read from 5' end to 3' end. Genetic code has distinct polarity, means it has specific pole. This end is called as 5' end, this end is called as 3' end, and always the genetic code is read from 5' direction to 3' direction. Now fourth characteristic is genetic code has a degeneracy. Means what? One or more than one codon are responsible for coding same amino acid. Means what? Just see, one or more than one codon. See, can you see this codon UUU? Another codon UUC. These two codons are responsible for coding same type of amino acid. These two codons UUA, UUG is responsible for coding same amino acid. We can consider these codons, these four different codons are responsible for coding same type of amino acid. So we can use the word genetic code has degeneracy, means one or more than one codons are responsible for coding same type of amino acid. Next, genetic code has dual nature. Actually this point is applicable for one codon only, AUG. AUG is playing dual nature, means it is acting in a two way. In first way, AUG act as start codon, means you can consider, if you are considering this as a genetic code, always towards the starting point, wherever 5' end is there, always you will get AUG. Means AUG is acting as start codon, and this AUG at the same time is responsible for coding amino acid, that is called as methionine. Okay, so genetic code has dual nature. It is applicable for AUG only. AUG is acting as start codon and it is also responsible for coding amino acid. Next one is genetic code is non-ambiguous. Non-ambiguous means it is specific. If particular codon is there, it will identify, it will code its own amino acid. For example, if AUG codon is there, it will identify, it will code methionine amino acid only. If UUU is there, it will code for phenylalanine only. Means they are specific in nature, or you can say they are non-ambiguous. Next one is genetic code is non-overlapping. Non-overlapping means what? See, genetic code is non-overlapping means each base or each codon, one codon is responsible for one amino acid only. I'll explain this with example. See, if this is genetic code, see, AUG will be the first codon. It will responsible for its own amino acid. UAC will be the second codon. It is responsible for its own amino acid. This will never happen like this. AUG will be the first codon, UGU second codon, GUA third codon. It is not like that. Always it is in the form of triplet. AUG means completely it is one separate codon. UAC means it is completely another codon. No overlapping is there. Okay, next is genetic code is universal. Universal means right from bacteria to human beings. If particular codon is there, if AUG is there, it is responsible for coding its own amino acid, that is methionine only. In all the organisms, right from bacteria to human being, if any codon is present, that is responsible for coding same type of amino acid in all the organisms. Here I had explained with example. If AUG is present in bacteria, it is responsible for coding methionine only. If AUG in human beings, it is responsible for coding same methionine only. Next one is genetic code is a triplet code. Triplet code means what? Genetic code is always in the form of triplet. This triplet is called as codon, and the codons are always in 5'3' direction, and each codon is responsible for coding their own or specific amino acid. Okay, so genetic code is triplet. This triplet is called as codon. Codons are always arranged in the form of 5'3' direction, and each codon is responsible for coding their specific amino acid. Next one is, last one is, genetic code has initiation codon and termination codon. Means what? See, if this is the genetic code, this is genetic code. This end is naturally 5' end and this end is 3' end. Always to 5' end is called as start codon. Whatever the codon is present here, that is called as 5' end, and whatever the codons are present here at 3' end is called as stop codon. Okay, now in all the organism, always towards the 5' end, AUG codon is present. Always towards 5' end, AUG codon is present in all the organism which is responsible for the formation of methionine. Means always methionine is the first amino acid, and always towards 3' end, stop codon is present. But here AUG is only start codon, but at the stop end or here at the termination end, any one codon will be there. It may be UAA, it may be UAC, it may be UGA at 3' end, and it is called as stop codon, or it is also called as termination codon. Why it is called as stop codon? Because it is not responsible for coding any type of amino acid. So here we have completed characteristics of genetic code.